something like that, but yeah.
So I'm going to get them fairly promptly, if that's okay, because people do things as sort of chop and change between sessions.
So I'll follow your lead.
It's just there's a lot.
Yeah.
Really good. I have been working at the EBI for the last 10 years. European Biobanks Institute, managing the industry program there. I recently left because of the rules they have about how we work there. Oh, really? Yeah. I'm supposed to work there for 9 years. Yeah. This is an intergovernmental treaty organisation.
Yeah.
It's a funny, it's not, it doesn't follow national rules or anything.
Okay, they set their own rules.
They set their own rules, yeah. Now I'm working part-time for Bristol Alliance Inc., which is a not-for-profit, global not-for-profit organisation. And again, with a, what is it? They organise pre-competitive projects to help the member companies who are pharma, science companies, software companies, also research institutes, to reduce the barriers to innovation in life science R&D and healthcare. And there's a whole range of projects.
I had no idea. That's awesome.
Bristol Alliance is a very interesting company. I can send you some stuff about it. Yeah. We have a lot of companies who provide services in this space are involved. Yeah. Okay, great. That's right. I'll send you some.
Yeah, do.
About 150 members, and lots. If you saw the list of members, you would recognise.
Yeah, it's just like a who's who in the zoo.
Okay, so good afternoon, everybody. Thank you for coming promptly. I'm going to try and keep reasonably on time because I know that people will want to sort of switch between parallel sessions, especially given you have this wonderful app and you can download everything and it tells you where to go and when. So my name's Dominic Clark. I'm the co-chair of the technology track for the International Society of Computational Biology. Very pleased to welcome our first speaker. It's David Hiatt from Weka IO. And David's going to be telling us about storage systems that support tomorrow's life science applications today. So, David.
Thanks, everybody. Can you hear me okay? I usually kind of have to adjust these things. So my job today is to put you comfortably and safely to sleep for the next 20 minutes since you're after your food coma. What I'm going to do is, actually the presentation's going to, I'm not going to do it in Swiss Dutch, or Swiss German, because actually my Swiss German is about as good as my Russian, which is slightly better than my Hebrew, none of which I speak. So today, I thought we would just talk a little bit about the importance of storage as it relates to discovery and the impact it can have on discovery. So the company is called Weka IO. We're based in San Jose, California, but we also have offices here in the UK and also Israel and Germany. And what we're doing is essentially we're providing a software answer to accelerate data-intensive applications like machine learning and genomics research as well. And we'll do this either in an on-premises in the data center or within the cloud environment. So being software, we can pretty much run anywhere we choose to. But let's take a quick look at the most common workloads that we see. Essentially are genomics and bioimaging, and actually they're quite diverse, quite different. They're almost polar opposites in that sense because they all have the same basic workflow in that you're ingesting information, you're also doing processing on it, then you're storing it for longer term, and then collaboration, you're sharing it over a broader network. But that's kind of where the similarities end from the standpoint that in genomic data, depending on whether you're doing exomes or whole genomes, the data sets are quite different in terms of size. The pipelines you're using vary greatly, and as you process the information, you're actually processing and you're reducing the amount of overall data. The value is in the VCFs and the variance. Whereas if you look at bioimaging, you can start off with, depending on the modality you're using, whether it's cryo-EM or some other microscope technology, you start off with very large files and they just grow. It's called data amplification, and we'll talk about that in just a second. But that is predominantly all unstructured data, and you're adding structured data to it through annotations. So in terms of that, from a storage perspective, things get pretty ugly very fast. In terms of genomics, massive text and binary files are commonplace, and you're looking at endless directory trees, and anybody who has actually tried to do a list or a find of a file understands how painful that can be, especially to other users. But ultimately, you're talking about billions of very tiny files, and traditional storage systems don't handle those well, especially if they're based on spinning disk. When you look at all of those open and closes that are occurring during the analysis phase, every single open and close has to be logged, and that's a metadata operation, and that too, those are like overhead. That's a penalty every time you do that. There's no added value to that activity, but it's really impacting your the pace of discovery if it slows down the access to the data. You look at the imaging data. Now you're talking about the detectors on these microscopes are increasing. They're much faster, and they're much higher resolution, and as a result, now you're looking at terapixel type images, again, that grow in size. And traditional architectures, you can kind of see there's an HPC-centric one on the top, where you typically have a compute cluster followed by some form of a SAN and block storage on the back end and a parallel file system fronting that. And in essence, these are very complex to manage and don't scale well in terms of overall growth, and they certainly don't have a connectivity into cloud, especially on or a hybrid type cloud solution. And if you look at the another common architecture is the NAS-based systems, traditional filers. Those could be the Cumulos, the NetApps, and so forth, Isilons that you may be familiar with. And in essence, the performance of these is limited by what a protocol, network protocol, it's NFS, it means network file share. And ultimately, that protocol's performance is dictated, and there's no improving it over what that limit is. So ultimately, you've got to kind of look in different directions for performance. And so if we step back just a second and take a look at what's happening on the infrastructure side, you see a lot more CPU cores on the CPUs these days. That gives us more processing power, but you also see a lot more use of GPUs. And GPUs have hundreds and thousands of cores inside of those and are great at parallel operations. The challenge is now we've condensed the compute into a very dense package, and we have to now transport all that data to the compute layer. And now we need a really fast network to do that. And again, that can be a problem, especially in large environments where you may be sharing the infrastructure as part of a central IT organization. Then you couple that with the data growth in life sciences. It's traditionally doubling every 7 to 9 months. And that causes a problem just from a processing perspective. And the drivers of that growth are obviously the collaboration and sharing of data that's required, but the new technology I already mentioned, it's faster. The sequencers are getting much faster. They're doing much longer reads, which is great. It helps with the computational side somewhat, but nevertheless, you're sequencing more. And then finally, you have data retention policies, and those could be either mandated, especially in the healthcare space, as to how long you have to keep that data, as well as funding mandated for on the research side and the academic side. And then now there's really value-based, and that could be indeterminate in terms of the actual time of keeping that data. And ultimately, that's driven by a lot of the new applications and related to AI machine learning. And then, of course, digital imaging is becoming quite popular. Cryo-EM is expanding, the use of that is expanding rapidly to support the genomic side, because you're visualizing things, and this is where that data amplification comes in. So let me just take a second to kind of explain that and give you an example. So on the left of this graphic, you see the actual slide, the sample in digitized format, and in the center and the right, you see all of the annotations that go along with that. So there's a lot of data describing what is on that slide, but that's not the problem. If that's a single focal plane, you're looking at, say, 15 gigabytes for a single plane, and you'd think if I need multiple planes to make a three-dimensional image, it should be 10 times that, when in fact it's 250 times that. So no IT organization has any idea that this is coming. When you get a new instrument, and I see this quite common in some of the private labs, they'll get a new microscope, and IT has no clue what's coming. And so that causes problems. So in essence, the infrastructure has to be flexible to keep pace with all of these. You have on the right, the graph you can see, this is actually from the protein data bank, and shows the number of proteins maps that have actually been submitted. And this is actually out of date. I think they have a new map with forecasts, and it's quite a bit larger than this, but ultimately it gives you that idea of the amount of research that's taking place and the technology that's driving some of this. AI is surprisingly, I thought that maybe retail and finance would be probably the most heavily used using AI machine learning, when in fact pharma and life sciences are. Couple that with healthcare is up there as well. So all of these have their own needs as it relates to storage. And so the demands are changing. People are looking for on-demand. A lot of small startups, they can't afford the infrastructure, so they're going to the cloud, at least in the early stages, until it gets too big and too expensive, and then they develop the architecture on-prem. And collaboration, data sharing, that's all pretty much table stakes. Everybody in research has to do that, and certainly even on the commercial side, if you're working with research partners. But then there's the standardization of data formats and metadata. Think about it, if you're collaborating with a colleague, are you describing the data in the same manner that are you using the same descriptors for that data as they are? And so you need alignment on that front. Otherwise, you end up with mismatches of descriptors. And then, of course, there's a lot, as I said earlier, the processing types are changing. The GPUs, now they have TPUs, in fact, which are tensor processing units, very specific to TensorFlow AI machine learning applications. Then you have FPGAs. Some of you may be familiar with those through the Edico Genome Genomics acceleration platform that Illumina has. But ultimately, these are all hardware things that don't scale well, and they have limitations. And really, if you look forward for flexibility, software is the most flexible and dynamic. And so the storage is really where the software is taking a leading role in this. And so what it allows us to do is kind of bend some of the rules that were inflexible in a hardware situation. The common thought is that I have all this compute, I need my data close to it. Actually, that's no longer true, because with the low latency networks I was talking about earlier, if it takes me through a direct connection 100 microseconds to write a 4K block to a SSD or some disk drive, I can transfer that same 4K block over a network, a 10 gig network, in 5 microseconds. So data locality doesn't mean anything, not what it used to. I can distribute that, and that's exactly what the software takes advantage of and allows us to do. So in essence, if I take that same 4K block and slice it up into bits and parallelize it and transfer it, I can transfer it in much shorter time, and that gives me a lot higher throughput and faster access to my data. And so that downstream effect is researchers aren't waiting. They have access to the results much faster. And so conceptually, if we go back to the architectures I was talking about earlier, we have the compute layer up top, and it can be either GPU or CPU based. You have the network shares out on the far right, whether it be Linux or Windows. But ultimately, the software resides on the same types of servers that are used above are used below. So there's standard commodity type hardware running software that turns that all the SSDs in those devices into a single shareable pool that now becomes your primary storage with all the advantages of one, shareability, but more importantly, the performance that it provides. And especially for the genomics data sets with small files and random I/O. SSDs are very good at that. Hard drives are not. So obviously you can't put everything on an SSD. It's too expensive to do that. So there has to be some other way to scale more cost effectively. And that can be done through tiering, automated tiering to a file system or a object store. Object store can be on premises in the form of a physical device, like a Western Digital Object Cloud system, or in AWS for the public cloud. Not a lot of people have access to and or can use public cloud systems. So the flexibility of having multi-targets, meaning I can send my data to an on-prem object store, or I can send it to a separate data center. Again, it's all private, but it's in a different location. Now I have disaster recovery capabilities already baked in. What does that look like from an application? Because you don't want to change your applications at all. Ultimately, all of the application sees the totality of those two. It doesn't know whether its data resides on flash or an object store. It doesn't care. It just needs access through a file system that looks, acts, and smells like a local file system. And that's what this particular software does. But it also allows you to support multiple environments. So, for example, I mentioned on-prem. Yes, it will run on-prem in the data center, but if I tier out to a public cloud, that gives me a hybrid type deployment, and I can use that for cloud bursting, which is essentially I can push the data to the cloud. I can spin up a cluster in the cloud 1,000 times what I could afford on-prem for a very short time, get my results, eliminate all the data. I don't keep the data. All I care about is the results. That's all I pay for when I pull them back. On egress, I pay the fees for the data results. And obviously, there's the full cloud implementation, as I mentioned, for startups and things. So I'd like to kind of shift a little bit to some newer applications. Okay, in terms of GPU acceleration, we have a partner of ours does GPU acceleration of standard genomics pipelines. And essentially, that provides the ability to accelerate the research process considerably. Another concern is obviously, all this data, what do I do with it? Where do I store it? It costs me a lot. And we've partnered with a company out of the UK called Petagene. They can actually compress, if you're familiar with CRAM files that are already compressed, they can compress it an additional 30 to 50%. So in essence, they can compress data up to 90%, which saves on storage, But more important than that is the pipelines run on the compressed file. You never decompress it. It's just run on the compressed version. So again, that's actually provides quite a bit of performance increase. And then finally, I'll just close out with a couple of customer cases where this is actually a very, very large implementation in the UK, where they're looking at, believe it or not, on flash of over a petabyte of flash. And they're going to scale that to about 5 petabytes on flash by the end of the project. And this is a large national genomic sequencing project. But ultimately, they have the other aspect, they're looking at close to 100 petabytes on the object tier. So it's a considerable amount of data, but ultimately they're all connected for the data resiliency and can share that data amongst multiple data sites. Not all people are that big. We have another customer, it is actually a smaller research organization, or research lab in a major university, and their problem is they're sharing HPC resources. And that impacts their research because being a shared resource, it delays their access and those resources get oversubscribed. And so from that standpoint, what they have done is they've actually moved to an all in the cloud type environment, which can scale up and shrink with their research needs. And of course that helps with their budgeting. But ultimately, what does it mean from a research perspective is they're getting 2 to 3 times faster performance than they were getting for their genomic pipelines, and the storage was three times faster than what they had in an on-prem deployment, and this is all in the cloud. So the concept of everything in the cloud is slow is not the case. Amazon has done a lot to build out that capability. And then finally, just IT, I hope that I've given you just a quick view of how important IT infrastructure is to the pace of scientific discovery. It does have a major impact on it. Life science data is different. It is probably the perfect storm of the ugliest workloads you could imagine, all mixed into one. And so it does require that flexible infrastructure, the ability to scale, the ability to access things in parallel, and then certainly the results are going to be that you can support those advanced analytics, the machine learning, those type GPU type applications, as well as improve the researcher productivity and ultimately speed up the discovery process. If anybody has any technical questions or would like to discuss it further, we have a table up in the exhibition hall. Happy to talk with you then. If there's any questions now, I'm happy to take those as well.
Thank you very much, Dave. So just very quickly, if anybody's got a question or a comment, thought, observation. Is this? I'm not sure. Because I don't think I can control this. No, okay.
I'll repeat the question.
Yeah.
So, my question, I was wondering, do I have to change my application logic in order to use your services? Let's say you put a directory in my HPC cluster. If we want to make a hybrid, something that works locally and as well in the cloud, do I have to change my pipelines, or they would just operate as if the path was in that particular area?
That's a good question. So in essence, he was asking, do I have to change my applications or my pipelines to take advantage of a hybrid type deployment? And the answer is no. From a standpoint of, say, I want to draw a distinction between the cloud-based aspect of the storage and the performance layer, that flash tier. That's where your analytics are going to be done. That's your active data set. That's where it's going to be processed. That could be in AWS. It could be on-prem. Doesn't matter. But that's ultimately the performance tier that you're going to have, and that's what your pipelines are going to run on. It's a file system. It looks like one. It looks like a local file system, despite the fact where its location is. Your applications do not have to change at all. But we will take care, we automatically tier, just to finish that thought, is that once you're finished doing the processing, you can push that data to the object store for more economic storage, longer-term storage. You can call it back at any time, but you save your flash layer for the active data and the active jobs.
Okay, so David, thank you very much for that
Yeah.
answer. Thank you very much for that question. Thank you very much for your presentation.